second speaker. Thanks, everyone. Welcome everyone to the second talk of today's seminar series. We have Maxim Zaslavsky with us today from Stanford. He's a PhD student there, and he's going to talk about machine learning on immune receptor repertoires. Very nice to have you here, and looking forward to your talk.
Thank you so much. Let me just make sure. Can you see my slides now?
Perfection, yes.
Okay, thanks for having me. I really appreciate the opportunity to be here. Hi, everyone. Good morning from California. My name is Maxim. I'm a grad student at Stanford, working with Scott Boyd, Anshul Kundaje, and Rob Tibshirani. Today, I'll be talking about our recent preprint called Disease Diagnostics Using Machine Learning of Immune Receptors. It builds on the phenomenal work that the folks in the field and perhaps here in the audience have done, so I'm eager for your feedback. Let's see. I want to start by putting our work in the medical context. Medical diagnoses rely on a wide variety of lab tests, and some of these tests are very specific. For example, detecting SARS-CoV-2 viral RNA is a direct signal of COVID-19, but other diseases are much harder to diagnose with today's tools. When it comes to a complex autoimmune disorder like lupus, for example, diagnosis often requires a combination of finding abnormal symptoms, finding autoantibodies which are not specific, and excluding other possible conditions. Altogether, that's quite the odyssey for some patients and can delay treatment. What's interesting is that the tools of diagnosis make little use of the immune system's internal record of disease exposures. In response to disease, the antigen-specific receptors on our B and T cells change in composition. So can we survey that immune repertoire to see what the immune system is reacting to? But I need to pause here and say, I'm not a clinician, I'm a computer scientist. And we all know there's a rich history of outsiders coming in and thinking medicine is backwards and we can do everything better. There's quite a graveyard of new technology methods claiming to revise medical diagnosis. So we have to be realistic when it comes to human health. Lab tests are just one piece of a bigger process. They need to be viewed in conjunction with the physical exam, with medical history, and with other medical contexts. So with those caveats up front, let's dive into whether we can read the disease exposure records from the body's own surveillance system as a way of contributing to the diagnostic toolkit that we have today. What makes this hard is the high variability of immune receptors across people. In our study, we looked at B cells specifically. So here's how B cells receptors develop in a nutshell. First, a random recombination of V, D, and J gene segments creates a diverse starting pool of naive B cells. Some will be stimulated by exposure to an antigen, like a virus or a vaccine. And then those cells undergo further diversification. That includes somatic hypermutation, so the accumulation of further mutations, and clonal expansion of those stimulated cells. This diversity adds up, and the result is that exact matches are rare, even in people exposed to the same disease. Also, even in patients with an active illness, only a fraction of their immune receptors might be devoted to the responsible pathogen. So we would need to find the rare disease-specific B cell receptors to decipher someone's immune status. What's our approach here? Well, we start by sequencing B cell receptor heavy chains from the peripheral blood of patients and healthy donors. And the way we've been thinking about it is that in healthy people, you predominantly will see background exposure sequences, which I'm showing in green here. Whereas in disease patients, sure, many B cell receptor sequences will be related to background exposures, but some will be disease specific. And here I'm using the blue color for that. Now, this is important. We don't know which ones are disease specific sequences. We don't have any training labels at the sequence level. Our only disease label is at the patient level. So what we know is this bag of sequences came from a COVID patient, but we're not told which needle from that haystack is COVID related. And that's what the machine learning algorithms we're introducing here will try to find to then pick up on COVID exposure signals in other individuals. In our study, we had 355 people with over 10 million B cell receptor heavy chain clones, and we saw that exact sequence matches were very, very rare. We applied a combination of 3 machine learning models to identify the presence of infectious and immunological diseases. They captured different aspects of human immune receptor populations in order to find those disease-specific patterns in B cell receptor sequences. The first model predicts whether a person's B-cell receptor repertoire differs from those seen in healthy individuals. And here we're looking at overall repertoire composition, so V and J gene segment usage trends and rates of somatic hypermutation to find signs of an active immune response. The second model identifies groups of highly similar sequences across people. Now, this is a technique developed over the years by many groups, including folks here in the audience. And the idea is to detect highly similar CDR3 sequences shared between people with the same diagnosis. Finally, the third classifier uses a broader proxy for receptor similarity as opposed to direct edit distance comparison. And the reason why is that amino acid edit distance may not be the best way to find similar receptors because ultimately these sequences encode complex 3D structures and small sequence changes can have a big structural impact. So we wanted to see, can we extract information from these B-cell receptor sequences that could allow us to find more loosely related immune receptors that may still have the same antigen targets? There's been some interesting work in the direction of alternate representations like K-mer frequencies and biochemical properties to describe these sequences. Here we took an approach called language modeling. We use a language model here called the UniRep, which comes from this George Church paper. And UniRep is a neural network trained to predict the next amino acid in a protein sequence. It learns the relationship between different sequence regions, like learning human language phrases and grammar rules allows us to anticipate the next word in a sentence. Under the hood, the UniRep neural network translates each sequence into an internal embedding, a vector of around 2,000 dimensions. And if those 2,000 traits let UniRep predict the next amino acid correctly, then UniRep has managed to extract some fundamental features that describe the input sequence. Indeed, when UniRep was trained on around 20 million proteins from many organisms, its internal representation, and by that I mean these vectors, they were shown to capture fundamental protein properties like structural classes. So we can use this idea to create a numerical description of each B-cell receptor sequence. But first, we modified UniRep to train it on B-cell receptor data specifically. We thought that by making a version specialized for B-cell receptor sequences, we might get even better feature representations for disease classification. So what this means is that we combine the global patterns that UniRep has learned from many organisms with the specific patterns seen in B-cell receptor variation. So we use the language model to transform each amino acid sequence, I'm talking the CDR1, CDR2, CDR3 sequences, into a numerical vector. And you can make a matrix of sequences and their vectors. Here, each sequence is described by those 2,000 numbers that come out of UniRep. And this is your new training data. Now, as far as training labels go, again, you don't know which sequences are actually disease specific, but you can label them based on the diagnosis of the person they came from. That's what I mean here when I say that the sequence level labels are noisy. That's what these question marks here in the diagram mean. And then we trained a classifier that uses those vectors to predict disease. The trick here is evaluating the thousands of unique B-cell receptors in a sample, first on their own and then in aggregate. So we call this whole framework MALADI. That's short for machine learning for immunological diagnosis. And these three models are blended into a final prediction of immune status. Now, these are all good models on their own, but combining them compensates for any blind spots that a single one of them might have. In our proof of concept, we can distinguish COVID-19, HIV, lupus, common variable immunodeficiency, which I'll refer to as CVID, and healthy states. And this is all done simultaneously, meaning we screen each sample at the same time for multiple acute and chronic infections, as well as autoimmune and immunodeficiency disorders. This approach scores 0.96 AUC in a cross-validation test on our dataset. And AUC here is a commonly used metric of classification performance. Let me just emphasize how this compares to the other modeling approaches. So for comparison, that second model on its own, the CDR3 clustering by edit distance approach that has been around for a while, that scores 0.82 AUC on this data, 0.82, as compared to the 0.96 that we're seeing when we add all three techniques here together. Notably, the cases where classification was less accurate is also where the model is less confident in its prediction. So on the right here, we've plotted model prediction confidence as measured by the difference between the top two predicted class probabilities. A higher difference implies that the model is more certain in its decision to predict the winning disease label, whereas a low difference suggests that the top two possible predictions were a toss-up. The left bar here is for incorrect predictions, and you see there was less difference in the predicted probabilities assigned to the top two classes, whereas the right bar is for correct predictions, which had more difference in predicted probabilities assigned to the top two classes. So what this means is that the model is more certain in its choices when it makes correct predictions, while its mistakes tend to be close calls. So 0.96 AUC, is that too good to be true? Shouldn't it be harder to find these subtle disease signals in immune repertoires? And we looked at this and we realized we need to make sure that demographic variables aren't driving our results. There's many ways that subtle demographic differences can shape immune repertoires. Repertoire composition changes with age. Disease prevalence definitely depends on age, and where you live determines your environmental exposures. So we asked, are these effects driving the results here? We changed the final ensemble model to control for age, sex, and race explicitly. First, let me show you the performance on the subset of individuals with known demographic data without adding those controls. And here we get 0.94 AUC. And then after controlling for age, sex, and ancestry, the score drops slightly to 0.89 AUC. This slight drop means that demographics have an effect, but only a modest effect on classification. And 0.89, that's a more reasonable value, and it's still better than the 0.82 AUC of the standard CDR3 clustering model before any controls were added. So beyond making disease predictions, we wanted to interpret the classification model to extract biological intuition. And that's why we designed MalID to be interpretable, not a black box. The model generates a ranking of sequences, meaning it tells us which receptor sequences are prioritized as disease-specific. So we asked, which immune receptors matter the most for classifying different diseases? And we confirmed that these prioritized sequences match independently discovered biological facts. So for example, we ranked all the sequences from COVID-19 patients with MalID, and then we checked which sequences match Charlotte Dean's CoV-AbDab database of antibodies known to bind the virus. On the left in blue are sequences that don't match CoV-AbDab. They have lower rankings than the sequences on the right in orange, which do match CoV-AbDab. So this tells us that receptors known to bind SARS-CoV-2 were highly ranked by MalID. And that's even though the model didn't know when it was trained that these are binding sequences. They just happened to show up in the patients in our data set, and the model determined independently that they were important signals of COVID-19. This concordance between automatically prioritized sequences and experimentally validated disease-specific sequences from separate cohorts suggests that MalID learns meaningful rules that capture some of the same biological knowledge that we've gained during the COVID-19 pandemic research effort. What are the other properties of highly ranked sequences? The sequences that were highly prioritized for COVID-19 prediction seem to come from V gene segments that are common in antibodies that bind SARS-CoV-2. And I'm indicating those with red arrows here. So here I'm showing ranking trends for sequences from different V gene segments. The V genes on the y-axis are ordered based on their median ranking from least disease-specific or lowest ranked at the top to the most specific or highest ranked at the bottom. Same for HIV, especially V434 here, which has been seen in people who make broadly neutralizing HIV antibodies. Now, V434 is an interesting one because in the lupus literature, it's also described as an important marker of the immune response. But in the case of lupus, the model pinpoints V gene segments that are less well described in the literature. We'd expect V434 along with 439 and 459 to be prominent since these are highlighted in the literature, but those V gene segments didn't show up prominently in this classification here. We think that what's happening is that V434 is not specific to lupus, but may be a general marker of several autoimmune disorders. So when the model is trying to separate the different diseases here, and there are several with autoimmune components, V434 isn't a helpful indicator of lupus for this problem. Last thing I'd like to share is that this framework allows us to visualize and track how the contents of someone's immune repertoire change in response to disease. We created a reference UMAP layout of the model's internal representation of sequences that it thinks are highly disease specific. Here, each dot is a sequence, and the sequences are automatically arranged into regions that correspond to different immune states. Now make this reference map a background in gray, and we can overlay new sequences by running them through the same model and the same UMAP transformation. So as a test, we downloaded the sequences for a number of therapeutic monoclonal antibodies for SARS-CoV-2, and 6 out of 8 of these antibodies were projected into an immune receptor region specific for COVID-19 response. That's the blue region at the top. This is exciting because it shows you visually what the specificity of an antibody is. You can see that these are COVID-19 specific. But 2 of the 8 weren't projected in the right place. And we think this is probably because we trained our model on noisy sequence labels taken from each person's overall disease type. So that means it's designed for overall repertoire level disease prediction accuracy, not to be perfect at classifying individual sequences. You can use the same approach to visualize a patient's repertoire over time. Here's a patient 5 days after COVID symptom onset. Now, just to be clear, they weren't included in the reference map on the left. And here at day 5, so far their immune state starts healthy in the green region there. Then by day 12, you see a COVID-19 response emerge. Now, I want to acknowledge this isn't perfect and we're still working on this. The blue here is not quite in the blue continent from the reference map, probably because in a patient you'll have intermediate confidence sequences, whereas the reference map is built only from the extreme super COVID-19 specific sequences. But with that said, we think this is still a really useful way to visualize an individual's immune system over time and take a survey of their immune receptor specificities. So to summarize, the idea here is that exposures to antigens shape the immune system's collection of B cell receptors. And we want to take a survey of these receptors and figure out your current exposure to disease. We applied a 3-part machine learning analysis framework to data from over 350 people from 5 distinct immunological states, including viral infections, an autoimmune disorder, and an immune deficiency. We classified them with high performance, controlling for sex, age, and ancestry. And to achieve this, the models learned disease-specific patterns within these highly diverse repertoires of thousands of unique sequences. So they managed to extract some meaningful biological signals. Because malady was designed to be interpretable, we can study the patterns of receptors believed to be specific to different illnesses. And we saw that the highest priority sequences for COVID-19 disease prediction matched virus binding sequences previously identified from other patients in a variety of studies across laboratories across countries. Finally, we also visualized immune response changes during disease progression, highlighting the potential of longitudinal immune repertoire monitoring. Let me end with an important caveat. This is a proof of concept. We only have 5 immune states and several hundred individuals here. So we'd like to test this on larger cohorts to show that this works for more than just these 5 immune states. We believe this framework for disease prediction and immune repertoire interpretation can be very powerful. Thank you. And I'd like to also give a big thanks to our collaborators and to the patients who we studied for this paper. I'd love to take any questions and would be really eager to get your feedback on this.
Thanks so much, Maxim, for this very nice talk. There is a question in the chat, which you can maybe see.
Yes, give me a moment to pull that up.
I can ask it. Hi, it's Brian. Maxim, did you look at the CDR3 model by itself and take into account kind of the sex, age, and did its AUC drop as well?
So I haven't done that test. I expect we'll see a drop there of a similar size, but I haven't done that test yet, so I can't say for sure.
Yeah, I would assume so, but wanted to check.
I really appreciate that idea, though.
And regarding the CDR3 model, is that the one that alone got already the 82% accuracy? Is that the one?
Yeah, that's right. So it comes in at 0.82 AUC, that CDR3 model.
And so what would happen if you used only this model to compare whether you can find already COV-ABDAP antibodies? Would you find already then the significance? So basically the question is, do you need all 3 representations to get significance for finding an enrichment of you know what I mean?
Yeah, I think my answer is, unfortunately, I don't know. But I think that I'm very encouraged by the massive jump from 0.82 AUC to 0.96. And so I think having this combination and having that ability for each model to correct each other will do a lot. But I haven't done that test.
But it
And
would be
if
nice,
it's helpful,
yeah. But it would be nice, I think, to see how much each of them detects, right? Yeah, that's just a suggestion.
Yeah, and if we can show that that significance level improves.
Yeah, right. That would be nice.
Right.
Yeah, that would be nice. Yeah.
Thanks for that idea. I'll definitely work on that.
Gur?
Hello, I have a question. I guess you cannot map these repertoire sequences, the individual specificities, onto the compartments of the B-cell repertoire, like isotype or memory or natural antibodies. From here, I see that if you can isolate the effect of the CDR3 clustering, if you're isolate the effect of the public antibodies, you may have some idea maybe, because I think the highly specific antibodies and the natural antibodies may have very different contribution and different meaning in this approach. We are trying to work to unravel the IgM repertoire in the natural antibodies and its capacity to predict, and we find some. And of course, it's not connected to highly specific antibodies, but still it has capacity to predict. So for me, it will be very interesting if we can differentiate between the compartments.
And when you say compartments, are you referring to, you mentioned isotypes and naive versus antigen experience versus memory cells up at the beginning of your question. Is that what you mean?
Both actually, but and the way they go collinearly, because the interesting compartment is the natural antibodies, which are supposedly not specific. And they are very diverse and they are more dynamic in their changes. The very specific antibodies, on the other hand, can be often specific for the individual, and as biomarkers they may somehow fail, although of course we find common variable region families expressed over and over for the same antigens, so this is there, but generally my question is about this, the pre-immune and the post-immune repertoire, because the pre-immune repertoire has different physiology.
Yeah, and you know, I think that there's more that we could do to study the difference in the contributions from those public clonotypes versus the private, unique to each individual ones, right?
Yeah.
I think I'm encouraged by the fact that, you know, the reason we went down this whole like language modeling stuff, you know, it's so complex. Why do we need all this? And the reason we went down that route to begin with is because we saw that each one of these models contributes to the overall diagnosis and to the prediction.
That's already interesting. Yeah.
Yeah, and so what that leads me to believe is that there is information to be gained by going beyond just looking at those convergent sequences. But as far as categorizing or like giving you a numeric value of just how much each one contributes, I don't know if I have a great answer. And maybe one thing that I could tell you to answer part of your question is, so far we focused this on antigen experienced cells. So we've removed naive cells that are unmutated from our experiment here. And the reason why, because we wanted to capture the ones that seem to have some connection to disease exposures, right? As opposed to just the, you know, starting pool of cells that then might be stimulated once you're exposed. So I don't know if that gives you the color that you were looking for.
Yes,
but
yes, definitely.
I think I appreciate the idea though let's see if we can address this public versus private clonotypes and their contributions question some more.
Thank you.
Thank you.
Then we have Gur, and then we have another question in the chat.
Okay, I have a couple of things. So first of all, very, very nice talk and very beautiful work.
Thank you.
The language model, I mean, is it supposed to capture the mutability patterns that in the V gene, or do you train it only on the CDR3?
Yeah, so in this case, what we've done is we've trained it on the CDR1, 2, and 3. And so it should capture the mutability patterns in the V gene. And but we think it's doing more than just capturing the V gene information. You know, the V gene information should already be part of the other models here to some extent. So we believe that it's taking into account a more global look. You know, I'm not here to advocate for that as like the de facto approach. I think it's worth trying this with a few different ways of maybe focus only on the CDR3 or focus on the entire VDJ rearrangement and see what happens.
Yeah. Okay. So that leads me to another point, which I think extremely important nowadays. It's like the batch effects.
Yeah.
So do you think that your representation, which is quite uniform, I would say, because you can take whatever new repertoires and put it with this, could they somehow correct for batch effects? Did you try different libraries and see what is the effect of them? Did you like, maybe, you know, you can somehow gauge the data by moving into the healthy cluster or something along these lines? I mean, bottom line, did you look at the batch effects in this representation?
Yeah, I think this is a really, really important question because this is kind of, this underpins, can you trust these results or not, right? The way I've been thinking about it so far, I'm curious what you think about this, is that there's these other covariates that definitely affect our data besides batch, and that's, you know, age, sex, and ethnicity. And I'm worried that those will have potentially much bigger effects than batch. You know, in this study so far, we've collected all of the data sets with the same sequencing protocol over about 10 years. And so my worry is more that you'll see massive repertoire differences if you have one cohort of, you know, people from predominantly one age group, one ethnicity versus another cohort from another disease type with a different gender and a different ethnicity and a different background. And I think that might completely overwhelm the batch effects here. So I approach this as being more concerned about those effects and trying to tease those out. I don't know if that's a satisfactory answer to your question, though. I believe there's always more to be done on testing, you know, injecting more batches into that healthy group. I think that's what you were getting at, right? And seeing, can we, are they confused here or are they being pulled apart?
Yeah, even moreover, I think, I mean, what you described as batches, like kind of biological batches, but indeed, if you try to combine different library protocols and different labs, you would see very big differences that I don't know if they are so little compared to the age, sex, and other things. So I would look at, you know, try to look for other types of sequencing and see how they behave in this kind of representations. And this,
Thanks.
And this brings me to the last point, which indeed, like other protocols. So another protocol which is getting more and more popular is single cell, which you have paired heavy and light. And I wonder how, you know, if with this method you can have somehow compare the signals between the heavy and light in a paired way. Or,
yeah. Go ahead. Sorry.
No, or extend the method to incorporate both heavy and light.
I think we could definitely do that. You know, my hope here is not to you know, insist on one particular design of you know, how we insert data into this framework, but more to present to you all a general approach for stack a few models together and try to pull out some biological intuition along with the disease prediction. And I think that extending this to paired heavy and light chain data is definitely possible. Let me add one caveat as to why we haven't done that, though. And that's because we were worried about, you know, so to get that paired heavy and light chain data, right, you would probably do a lot of single cell sequencing. And that gives you a dramatically different amount of data, right? To get to the same level of data set size and repertoire depth that we have here, which is you know, around 10 million unique B-cell receptor sequences for these 350 patients, that would take a lot of time and a lot of money and would be very cost prohibitive. So we decided from that perspective that yes, single cell could give you a lot more information, but when it comes to finding these overall disease specific signals, I think that having more patients and seeing more of their breadth of their repertoires might have more value for this problem. Ultimately, if you were to use this, I think it would be very smart to bring both together, to have the bulk repertoires to sort of feed the model, and then to reinforce it with this maybe more limited set of paired heavy and light chain data. I think that sort of flexibility could be easily achieved in this framework.
Okay, we have 3 more questions, and in the interest of time, let's keep the questions and also the answers short and concise, please. So the
Got
next,
it.
So the next question is in the chat by Savita. You can also read it.
Yeah, I can read it out loud.
Yeah.
Thank you, Savita. So the question is, from your experience with different disease models, what percentage of the repertoire is disease specific, and did you see it differ between diseases? I think this is a brilliant question, because this is kind of underpins the assumption we've made here of, usually these disease-specific receptors are so rare in the repertoire. How do we find them and pull the signal out? I don't have a great numerical answer for you of the percentage, but I think that what this is showing us is that once you've done this, first of all, this filtering down to the antigen experienced clones, so you remove the naive B cell compartment, and once you try to prioritize sequences with this kind of a ranking, the disease-specific ones seem to pop out. So I would give you a rough, just back at the envelope estimate of around, 10% is the way I've been thinking about it, but I don't have a numerical way to prove that to you. Maybe I can give you a better answer to this in a few months.
Then we have Yuval, who raised his hand. So Yuval, please ask.
Yes, I have a quick technical question. I see that
Please.
Can you hear me?
Yeah.
Yes, go ahead.
Oh, sorry. So I have a quick technical question. In the language representation, if I understood you correctly, you're using the approach from the paper by George Church and his group, and yes. It's kind of trying to predict the next amino acids, right?
Mm-hmm. That's right.
It's a forward type of thing. There are, as you know, many other models like BERT and others that can go back and forth, and there are even richer representations for sequences. So I wonder if you try to play with this part of your algorithm and see differences. And just for curiosity, why do you think that the model from George Church can predict actually structure if it goes only forward? But so try to answer the second question is whether whether other methods of representation showed any difference.
Yeah, I think that's a great idea. Thank you so much for that. We should definitely try other language models there in the future. I think I'm encouraged by how high the performance is already. And that's sort of why we decided, we don't need to try any of the other ones just yet. This is enough to extract some biological insight. But by no means does that mean that UniRep is the perfect one. And I think I'd actually like to go try some of those other ones like ProtBERT and the other ones that you're saying are more of like a transformer structure and have a richer representation than just let me predict the next amino acid in the sequence. And I think they've been shown to predict structure and things like that even better than UniRep, right? So I'm not surprised that taking a deeper approach will extract more. What is a surprise is that this works already. I was expecting this to require a lot more fine tuning and a lot more richness of the representation, but it looks like stacking these, and you could probably even add more here, right? This is a flexible enough framework to add additional models or swap out any one of these components and train a new ensemble of models and then interpret it. So that's our hope in terms of next steps. Keep refining these, keep seeing if we can get better representations and learn even more.
And one last question in the chat, if you can read it again, Maxim.
Definitely. What is the data source of the training data set used here? Do data sets from other labs work well with this model? I think that's a two-part question. Let's start with this first part that I just read. So the data source is all from Scott Boyd's lab at Stanford, where we've run the same sequencing protocol for about 10 years now. And that's helped us a lot in terms of uniformity and reduce some concern about batch effects. But to be clear, there's still samples coming from different collaborators, different sources across the world. And they're including healthy controls from each one of them. So there is definitely a mix of batches, even if it's the same protocol. I think this will work on outside data. I think that you'll see that the features that we've learned here will translate. And I would love to go and try this on iReceptor data and see what happens. Second question, does the BCR fine-tuned model outperform the existing UniRep? Yes, that's how we ended up creating this fine-tuned model. Is first we scored it based on the original unsupervised task that UniRep tries to get very good at, which is predicting the next amino acid. And we saw that the fine-tuned model does a much better job of reconstructing the amino acid sequences for B cell receptors in particular. And then when we ran it through this pipeline, we saw that it performed well on the disease classification test. And I think that's generally accepted as a good way to use these language models. You take an off-the-shelf one, and then you let it capture those global patterns it's learned from proteins at large, and then you specialize it to your domain and try to make sure it can master the variability of your B cell receptor sequences in particular. Thank you so much for the questions, folks. Any others, or happy to also take this to Twitter or the internet, of course.
I think we're at the end of this beautiful session today. Thanks again to the two speakers, both fantastic talks on TCR antigen binding and also on machine learning of immune receptor repertoires. And with that, we close today, but we will be back in August, I think, at least. Thanks, everyone. Bye-bye. Bye-bye.
Thank you so much.
Thanks so much. Bye-bye.